So first of all, also thanks to Twist for inviting me to speak today. So I'll do my best today to provide a bit of an overview of some of the research that we have done in this field of circulating tumor DNA analysis and how we've utilized the new Twist cell-free DNA library chemistry to improve our detection of somatic mutations for various clinical applications. So I think a first good place to begin with is just to talk about, in general, cancer biomarkers, which I guess in general terms is a substance or process which is indicative of cancer in the body. And importantly, in oncology, it provides a critical point of consideration when we're talking about the clinical management of cancer patients. And it can provide prognostic information and important diagnostic information about the patient's treatment, but also how well the patient will respond on a particular drug. It can tell you about the pharmacodynamic processes that are occurring based on a particular treatment type, and if provide understanding of when potentially when a cancer reoccurs following treatment. Now, as cancer marker, we have many forms that are clinically approved today that are used worldwide. And I've just shown a few common types of that in this particular slide here. For example, those that are tumor-based are immunohistochemistry as a good example. Shown here some common immunohistochemistry stains used commonly in breast cancer, which provide prognostic but also predictive information about certain types of drugs that can be utilized following analysis. In this day and age, we also have the advent of next-generation sequencing approaches, which is able to provide direction in terms of selection of targeted therapies for patient care. Now, cancer biomarkers can also be non-tumor based. For example, on the top right, you can see that we have many forms of radiological imaging, shown here, for example, as PET scans, which are able to absorb glucose uptake and provide a spatial map of where the tumor is located. Importantly, there's also a number of range of these so-called liquid biopsies. Now, these have been used previously, such as things like alpha-fetoprotein. But it's not surprisingly that in the last few years, the use of these non-invasive liquid biopsies has been revolutionized in terms of their growth, particularly in line with the advances in genomic-based technologies. So what are the differences between tumor biopsies versus liquid biopsies? Well, first and foremost, tumor biopsies provide histological evaluation and provide a standardized workflow for evaluation of the tumor itself. For example, molecular pathology testing. However, it has several disadvantages. First and foremost, it being very invasive procedure. Generally speaking, it can also have a very long turnaround time in testing, particularly if, for example, the tumor sample is from an external pathology lab. And because you're only sampling one area of the tumor, you may not capture all the molecular features from all sites of the patient. Hence, you're only providing a small window of the actual tumor itself. Liquid biopsies, on the other hand, are obviously non-invasive, can also be frequently collected and analyzed serially throughout patient treatment. But because you are capturing this reservoir of all tumor sites, you're potentially providing a more holistic profile of the patient's cancer. One of the main disadvantages of liquid biopsies is that because this is the amount of tumor material that is floating as released into the bloodstream, for example, is only very minute, you need very sensitive methods in order to detect this liquid biopsy analyte. Now, liquid biopsies can take many forms and come in many different shapes and sizes. For example, they can be collected from many different bodily fluids, such as cerebral spinal fluid, saliva, but the most common one being a routine blood test. In terms of the analytes, there are many that they've studied in the literature. These include things such as exosomes, circulating microRNAs, circulating tumor cells. But the one that I've, particularly my lab, has focused on in the last few years is that called circulating tumor DNA. So what is circulating tumor DNA? Well, we know that in the human body that each cell actually releases these cell-free DNA molecules into the bloodstream. These are often very short in size, about 170 base pairs in terms of fragment length, and have a very short half-life of about 1 to 2 hours. And importantly, we know in cancer patients, they also release their cell-free DNA into the circulation. Now, because the circulating tumor DNA carries the same mutations as that derived from the tumor, they can potentially act as a personalized biomarker for cancer patient management. Now, as I said before, because the amount of circulating tumor DNA is quite low compared to the release of all other cell-free DNA molecules from normal tissue, you need very sophisticated diagnostic methods in order to detect this circulating tumor DNA in the blood. As a consequence, we apply many different genomic-based techniques in order to analyze this circulating tumor DNA. Some common ones include things such as digital PCR, which are able to analyze one mutation at a time. But it can also range from things such as targeted sequencing-based approaches, which I'll talk about later on today, as well as whole genome sequencing approaches. Importantly, it's always a bit of a balancing act here and a trade-off between increasing sensitivity, but also increasing genomic coverage as well as cost. Importantly, we get two critical bits of information from this type of analysis. One is a quantitative readout of the ctDNA, and this provides a measure of the disease burden or tumor burden in the patient, and is very useful in terms of tracking disease throughout a patient's treatment. The other bit of information that we collect is genomic characterization of the patient's cancer genome, and this provides critical molecular information and around potential clonal evolution and also potential resistance mechanisms. In terms of the clinical applications of ctDNA, well, this is one of the most exciting things about this field in that ctDNA potentially has utility in multiple facets of cancer patient management. And I've shown this slide here, which shows that not only can ctDNA be utilized right at the beginning of a cancer patient's journey in terms of cancer screening and diagnosis, but it can be utilized in terms of risk stratification, minimal residual disease detection, molecular profiling upon treatment selection, tracking of disease, and also detection of resistance. And over the next couple of slides, I'm going to show you some of the publications that we've done in order to explore the potential validity of these clinical applications across certain different tumor types. So the first one is around breast cancer, where we published this study previously a few years ago, which was led by one of my PhD students, where she was able to conduct next-generation sequencing on ctDNA samples from metastatic breast cancer patients. And overall, we were able to identify a multitude of different actionable mutations which had potential clinical benefit for patient care. In particular, we highlight this one patient where we were able to identify copy number changes in HER2 that arise throughout treatment, and this patient received anti-HER2 therapy and responded well to therapy. The next study that I want to highlight is one involving a patient who had lung cancer, where at the time the patient was enrolled in a clinical trial looking at a selective RET inhibitor. Interestingly, when we utilized the next generation sequencing approach of tracking the plasma samples throughout treatment, we were able to identify a multitude of solvent front resistant RET mutations, which were a clear sign of resistance to the selective RET inhibition. And these were subsequently confirmed upon rapid autopsy analysis of the samples via next generation sequencing approach. Overall, these kind of studies highlight the utility of using genomic analysis via ctDNA to understand not only molecular stratification of particular important targets, but also clear understanding of resistance mechanisms and clonal evolution of cancer patients, which was not fully achievable through just simple tumor sequencing alone. Now, for the rest of the talk, I'm going to move focus onto one of the more recent kind of projects that we've been working on in my lab, which is around glioma. Here, glioma is one of the most devastating cancers that we know about, with a dismal 5-year survival rate of only just 5% with very few treatment options. Now, standard care treatments include things such as surgery, radiation, and temozolomide chemotherapy. However, a number of different new investigative agents are currently under development. Now, like other cancers, glioma has a very well-defined mutational profile, And there are a number of different therapeutic targets that are now being exploited for therapeutic advantage, such as IDH1 and IDH2 inhibitors. But this underlines the need for the urgent need for new biomarkers which are able to direct these targeted therapies and provide disease monitoring strategies, for example, understanding of new treatment resistance mechanisms. In the context of ctDNA, unfortunately, CT analysis of glioma has been very challenging. And in fact, previous studies have shown that the detection of ctDNA in glioma is very, very low. It is postulated that this is probably due to the blood-brain barrier and the inefficiency of ctDNA molecules able to escape it into the peripheral circulation. As a consequence of this, other reports have been noted where there's now use of other bodily fluids, such as cerebral spinal fluid. And this paper in Nature that was published a few years ago highlights this approach. Unfortunately, while this does provide a very clear window on the clonal evolution and tracking of new mutations throughout treatment using cerebral spinal fluid. Cerebral spinal fluid collections are often not standard of care procedure and also cannot be frequently done throughout treatment. As a consequence of this, we wanted to see if we can then go back to the blood as a potential non-invasive source for molecular profiling. So this slide showed here's a recent study that we performed in collaboration with our collaborators at the Royal Melbourne Hospital, led by Associate Professor Andrew Morikoff. Here, we utilized a commercial off-the-shelf next-generation sequencing ctDNA panel, which covered 77 pan-cancer genes. And we were able to analyze a number of patients throughout temozolomide therapy. Interestingly, we were able to detect some key driver mutations through this approach, including IDH1. But importantly, we noted very high degrees of heterogeneity. For example, we only were able to detect 22% of mutations that were common between tissue and ctDNA samples. Importantly, we were able to identify through serial analysis multiple putative mechanisms of genomic resistance due to temozolomide treatment. This study also provided us the basis that there will potentially be future improvements to the technology, including the need to have a developed glioma-specific gene sequencing panel, but also an improvement to the underlying technology for ctDNA analysis in blood, as even though we saw an improvement in terms of detection of up to 40%, there was still plenty of room for improvement. So this leads me to the aim of our kind of study here, which was to develop a glioma-specific ctDNA panel. And we wanted this panel to cover several classes of mutations, including SNVs, copy number changes, and structural rearrangements. And the idea behind this panel was to apply it to various clinical applications, including mutational identification, utilize it for disease monitoring purposes, particularly throughout clinical trials, and also understand clonal evolution and detect mechanisms of resistance. This is where we turn to the utilization of Twist's hybridization chemistry. And we were very fortunate enough in terms of timing to also examine the new cell-free DNA Twist library chemistry and compare the standard chemistry as a head-to-head. In order to perform this, benchmarking exercise, we ran a number of reference standards as a matter of comparison. And these included a cell line admixture, which consisted of cell lines mixed at certain percentages with no mutations ranging from 0.1 to 4% VAF. This sample, which we will now call EF, also has a number of known wild-type locations up to about 100,000, which we know were considered non-mutated. We also ran a number of known Sericare ctDNA commercial standards ranging from 1% down to 0.1%. And for all standards, we ran them at 10%, but for the 0.5% VAF standard for Sericare, we ran it on varying degrees of DNA input, including 5, 10, and 20 nanograms DNA input. So what did we see? So this slide here summarizes the kind of the performance metrics of our analysis with the data on the right, shown in red, from the standard kit versus the data on the data on the left from the the standard kit versus data on the right from the cell-free DNA kit. And in terms of the total reads per sample post sequencing, we observe a pretty similar amount of total read coverage for all samples, maybe slightly higher for the cell-free DNA group. Interestingly, however, while the number of percentage of mapped reads was very, very high for each sample, when we look at the percentage of duplicates, despite the high number of reads in the cell-free DNA-based kit, we actually observe a lower degree of duplication, indicating their potential better conversion of unique molecules using this kit chemistry. Interestingly, when we looked at the evenness score, this was also maintained between both kits and was relatively high, above 0.8% for most samples. So given that this kit's advertisement point was the fact that you can get a high conversion of unique molecules, which is particularly important for cell-free DNA analysis, but also at low input, we also wanted to see how much this translated in terms of UMI replicate range. And when we compared the standard kit versus the cell-free DNA kit, we nicely observed that there are lower replicate rates in general as compared to the standard kit for the cell-free DNA chemistry. Interestingly, when we looked at the general overall mean coverage of each sample, we also see a higher observable mean coverage for those samples that utilize cell-free DNA chemistry. And in particular, when we went to 20 nanograms, we observed a very high difference between the kit, indicating a much more better conversion rate of unique cell-free DNA molecules that are sequenced. So when we then moved on to analyzing the variants that were analyzed post-sequencing, we noticed a very common trend in that we observe a rough limit of detection of about 0.5%, despite the different kits. But what was really interesting to observe was, in particular, those that were at high inputs, for example, at 20 nanograms, we observe a much better recovery rate of known positive mutations. Interestingly, we observe a common true negative rate. However, we did get one false positive using the new cell-free DNA chemistry, although this was a borderline case due to an indel caller in our evaluation. In terms of the EF standard, we see very similar rates of detection of variants between the different kits. However, we noted that this was likely due to the low DNA input, and we're currently evaluating high inputs to see if we can see if there's a significant difference between the kit chemistry when a high DNA input is utilized. Although noting that the false positive rate for both of these kits was very, very low in both instances. So overall, I hope I've provided some basis around how we can utilize liquid biopsies such as ctDNA to provide a non-invasive cancer biomarker for various clinical applications, how we can utilize Twist's cell-free DNA library prep kit to provide a sensitive approach for mutation detection, particularly with low amounts of DNA input. And our future efforts are now aiming to apply this panel for further clinical samples and test the clinical validity of our panel for future studies. And with that, I'd like to acknowledge members of my lab and all our collaborators, particularly from the Royal Melbourne Hospital and WeHi, who have helped with this project. Thank you very much.